Cool. Thank you so much. Okay, we're live, right? So hello everyone. My name is Lyona Carter. I go by Metalee in the Metaverse. And today I'd like to welcome you to a space that I'm centering around Henrietta Lacks. So I customized this space to teach others about her significant contributions to the world, not just to science. Everyone thinks, oh, biotech science is therapies. She contributed to the entire world, and very few people, way less people know about her than I'd like. I'd like for everyone to know all about Henrietta Lacks. So today I titled this presentation, Medical Contributions of Henrietta Lacks in Biotech, Holding Space Within the Metaverse. Again, my name is Metalee, and my background is I'm a clinical data manager by profession. I actually manage clinical trial data. I'm the person that polices data, and I make sure it's safe and protected and curated well, clean, honest, and accurate. So this is what I do. I've been doing this for over 20, 25 years. Actually, I want to say between 15, 20 years. But I've been in academia for over 25 years. So I've also had a stint as an extern at the CDC. I actually could read chromosomes and karyotype by the age of 17 because I was a bit of a science geek in high school and won a spot in the New York Academy of Sciences as a young scholar and was able to work in labs at age 15 and 17. In my last lab stint, I actually learned how to read chromosomes, which is called karyotyping. So I went to college with that. But what I'd like to do is refocus our attention back over to Henrietta Lacks. She's someone that I admire tremendously. And as we start, I have a poem on the wall. It's a section of On the Pulse of the Morning by Maya Angelou that I'd like to read just to ground the space a bit. So I'm going to shoot myself over there. You're free to join. You can stay where you are if you're in the room. But on this poem, this section that I could pull in that would fit on the wall legibly, it reads, "Here, root yourselves beside me. I am that tree planted by the river, which will not be moved. I, the rock, the river, I, the tree, I am yours. Your passages have been paid. Lift up your faces. You have a piercing need. For this bright morning dawning for you, history, despite its wrenching pain, cannot be unlived, but if faced with courage, need not be lived again." And this is just a section of Maya's poem. It's called On the Pulse of Morning, and she read it at the inaugural I think it was the second inaugural of Bill Clinton, former President Bill Clinton. So yeah, that's a section of the poem that resonates with me, especially when I think about Henrietta Lacks and her contributions to biotech. This is a unique space in that it's virtual 3D in the metaverse with me as an avatar. And on the slides on your virtual laptops, I've placed some facts about Henrietta Lacks that talk a little bit about who she was. She was born August 1st, 1920, as Loretta Pleasant. She changed her name later in life to Henrietta Lacks. Her birthplace was Roanoke, Virginia. She's lived between Roanoke, Virginia, Clover, Virginia, and also Baltimore, where she worked. She was a tobacco farmer, the mother of 6 children, 6 wonderful children, of course. She was diagnosed with cervical cancer in 1951, so she was very young. She was not even 35, so she was born in 1920. She passed away in 1951, and it was Dr. George Otto Gay who diagnosed her with cervical cancer. Henrietta was very smart. Like, she went to the institution knowing that there was something wrong with her and convincing the staff that there was something wrong with her, and she can explain what she was experiencing to a level that I think very few women did at the time. Like, she self-examined herself and knew that something was off, and she had the courage to get up and say, hey, something's not right, I need help. And she went and presented herself to Johns Hopkins. What we forget in 2022, what we can forget, is very few hospitals and institutions treated Black folk. So you would show up and you could get turned away. Johns Hopkins was one of the few places that provided us with health care if you're African American or African descent. That was one of a few places we could actually go and receive care. So Johns Hopkins Hospital is quite pivotal in this discussion. Dr. Gay was looking for years. I think he had been looking for over 20 years on a cell line that would live, because most times cells died under the hood. The hood is the lab bench, and when you're working on the lab bench, they used to call it the bench or the hood. So that's just lab speak. So he was working for almost 2 decades, if not more, looking for cells that would live past a few hours and days. Sampled Henrietta Lax's cells and found out her cells were quite just novel in the fact that they would regenerate in about 20 to 24 hours every day. So she has a remarkable cell line. They called them the immortal cell line. Her cells have just been pivotal to all sorts of groundbreaking innovations and help scientists understand protein expression, gene expression, synthesis, everything. I want to jump over to some highlights, and these are just highlights, guys, because nothing in this space can give credence to who this lady was and her significant contributions. I could go on for days just admiring her contribution. But some things that I've highlighted were her cells supported over or are referenced in over 11,000 patents, U.S. patents. Okay, that's significant. That's a lot. Her cells are called HeLa or HeLa, and it's the first 2 initials of her first name and the last 2 initials of her second name, Henrietta Lacks. Yes, and you will find, you could go to Google Scholar, you can actually do a search and see how many publications reference HeLa cells. Without HeLa cells, a lot of the therapies that we have and receive, we would not have. Before I go into that, I'll say they've even sent her cells up to space. She died in 1951. They started reproducing and submitting her cells to different places, and by 1952, so less than a year after she passed away. Unfortunately, she wasn't compensated, nor did she consent, which was quite the practice back then. They didn't ask people, hey, we have your cells and we want to sample them, or we have a sample, or a swab or a piece of tissue and we'd like to sample it. And they didn't ask for permission, they would just research it. So that was common practice. It doesn't necessarily mean it was the correct practice, because it wasn't, but that's what was common at that time, unfortunately. I do believe that at some point it should have been made known that this was happening quite early on, after it was happening, so that the family would be aware of it earlier. So outside, her cells have also been up to, I might have mentioned spacebound. Henrietta cells were sent up to space. They wanted to understand what zero gravity did to the human cells. I mean, like, we would not have the advancements in space and health research without her cell line, because they were able to actually take them up to space and look at them from space to see what the impact of zero gravity would do to them. So I want to talk about some of the supported therapeutics related to Henrietta Lax's contribution to biotech. I'm actually in the biotech field, started out pharmaceuticals, then went over to biotech, diagnostics firms, med tech firms of that sort. One of the highlights, without her, we would not have a polio vaccine. Like, think about that. I don't know if you guys know the statistics on polio and how contagious it was back in the day. You'd wake up one day and you could be immobile. You'd have all sorts of negative health effects related to that condition, that disease. Her cells were directly pivotal in relationship to finding a cure for polio, which is quite devastating and painful and impacted the globe. Okay, her cells are used today in HIV therapeutics. So the therapeutics, the antiretrovirals that are on the market today to treat patients with HIV, they've created those therapeutics with the support of HeLa cells or HeLa cells. Today, in our sad situation globally that we're dealing with, a global pandemic, COVID, we would not have a COVID vaccine without Henrietta Lacks. I could go on. I mean, you have no idea. There's other cancer research. There's other therapeutics. So when I support clinical trials, I'm in the rare disease space. I'm in the oncology space. Every space that I've been in, HeLa cells have been pivotal in the creation of novel therapeutics. So one of the things I wanted to consider was cells cloned in 1955. They actually I'm sorry, I can't hear. Is someone asking a question? Oh, okay, I'm good. I'm going to continue. Considerations, thank you. Considerations. So I'm bouncing back and forth between past, present. Bear with me, guys. Cells started being cloned in 1955. Again, no consent, no compensation, and the family did not learn that these cells were cloned until 1973, which was 3 years after Dr. Gay passed away. I believe he did not share with the world who she was until after but basically when he was sick, when he died himself of pancreatic cancer in 1970, but it was around before he passed that he shared who this lady was, Henrietta Lacks. He referenced her at that point, but the family didn't find out about her use until 1973, which is like 22 years after she had passed. 21, if not 22 years after she passed away. I have some pictures on the wall of Henrietta Lacks. This is her with her husband, David Lacks Sr. They had 6 kids together. So she's as you can see how beautiful she is. She's quite striking. She was known to have just a beautiful way about her. Her family calls her Aunt Henny or Henny. That's what they called her for short. I just think she's remarkable, guys. I guess you can tell. But on the wall over here, that's actually those are anyone's cells. Those aren't her cells per se. But I used to karyotype, and that's a picture of what cells, what DNA looks like when it's being captured. Like for example, when I've read them, I got a black and white copy of those cells to figure out which ones they were. So the top of it is called a P arm, the bottom is called a Q arm, and then there's all these, differences. That picture doesn't show it, but there's differences in sizing and lines on them that tell you which ones they are. And every one of them has a different characteristics that makes us who we are. I want to take you guys to this picture. You have to get up close to it. This is a profile of her cells that I pulled from one of the public DNA databases, and it's at the top. It's very hard to read, which is why you got to be so close to it. But at the top, it talks about HeLa cells. It talks about the 2,427 genes with a high-low expression, and what this profile is. This is pages. There's pages of this. If you go to this website, you can type in HeLa and you can type in BioGPS. A cell line, and you'll get this. It's publicly available. But you see these symbols. These are actually genes, and we get values from these. So this is what tells us a lot about what's going on with therapies and how it relates to what genes and all this wonderful stuff. So a lot of things happen with protein synthesis, and this is the data behind it. This is something that I manage as a person that manages data for clinical trials. So this is part of my wheelhouse. And the other pictures of the colorful pictures are actually pictures of HeLa cells in different processes. And I'm not a cell biologist, and so I won't pretend to be one, but I I actually love these pictures. But this is what HeLa cells look like. And if you see the first, I don't have the first picture of HeLa cells on the wall here, but if you go find what the first picture that Autogay took of her cells, they look like out of this world. They kind of scare me because it's black and white, and they got a lot going on. I'm just be honest. So they're not up in this wall. So I got the flat picture with the colors to help us deal. But her cells are wonderfully powerful. That other picture over there, the pink with the pink and yellows, I'm going to jump over. This one, let me back up. Yeah, this picture, let me stand over here. Oh, I can stay still. I'll stay still. Let me move again, and then I'll stay still. My bad. So this picture, if you look at the cells, you can actually see at the very top, you see that yellow touching the yellow? That, to me, I think is where it's regenerating. So you see where it's splitting, like where the yellow meets itself in the center? Yeah. I think that's where regeneration is happening. So this is beautiful to me. Like, I can hang this up on my wall. In fact, I have it in my Metaverse home because I think it's beautiful. Whoever took this picture, I pulled it off of NPR, so credit to NPR for the images. I'm quite grateful for these captures. Yeah, so does anyone have any questions?
These are electron microscope pictures, are they not?
I think so, yes.
Oh,
yes.
Yeah.
And there's probably
like
some more things going on. Yeah, but it's certainly,
an electron micrograph.
Good. Definitely. Yeah.
Boy,
And
beautiful.
I'm not, I don't know I used to stain. Sorry. Go ahead.
Yeah.
My bad. The saddest thing, I don't, I know that what I know is that when they were informed, they were bothered by it. I think the family was, basically part of it was the family was going for their own health care, and then they were getting sampled themselves. And they didn't know why they were getting sampled themselves. So the researchers at the hospital were trying to figure out if this was something that was inherited by maybe, relatives of hers. And so I think I don't know the full story, but I do know when they were presenting to medical establishment at that hospital, there was things going on that probably kind of sent up a red flag. But then they were told about it. They basically were informed. And I know from listening to the stories that when they were informed, it just blew them away. Like it didn't sit right with them. They were thinking, my mom passed 20 years ago, 24. Like, how could she be alive? Because to the world, the HeLa cells live, and this lady passed. And most cells die after a certain while, but hers to this day regenerate. Like today, they're regenerating, right? Companies are profiting from them. The family is not equitably compensated, and they should be. That's my opinion, and I stand firm on that, and I will say it publicly to anybody, because I know what they make off of it, and the family should have a right to this stuff, to all of this greatness, rather, not this stuff, to this greatness, to this legacy, to this contribution, and that's not asking for much, right? That's their right. But yeah, I know that when they learned of it, they were floored. They were just floored. So. They actually launched a case this past fall, 2021, going after a few companies. I know most notably it was Thermo Fisher Scientific that they're targeting, because I think they make at least $1 billion per year off of HeLa cells, and that's not including the other companies and their therapeutics. That's not I'm not surprised, because I'm bothered. There's a part of me that's like, come on, let's be real, let's be correct, let's provide some form of remuneration for what this lady has done for the world. I mean, we would not be where we are. I wouldn't have a job. I mean, because my field wouldn't exist, right? I mean, if you want to be straight binary with it, if then else, if she didn't exist, I could not professionally go into that space and be who I am. So it's not even the patents, and it's not just the patents, it's not just the contributions to therapeutics, it's also that lots of lives. She's benefited the employees of these companies, the retirees of these companies, the chairmen and women of these companies, and their children. She sent other kids' children to college. Who's sending hers and her grandchildren, her great-grandchildren's children to college? So, that's how I feel. I'll get off my soapbox. And I thank you all for joining me.
Oh, I was going to say, I know you have to go, but after all of the, the coverage she's been getting recently, the documentary that was made, has there been any kind of motion to make sure that her family, is being cared for in some manner? Is there, I know now we probably have some laws to protect. I know we have laws to protect from that, yes. But yes. Are there ways yes. that it's still kind of nefariously extracting from within our communities that you know about, that we should be cautious about?
I will say where I sit, I safeguard, right? So I'm a data steward. I protect data. I protect patients. That's my job. I'm unbiased on teams. Like, I'm biased towards the patient, but I'm not biased towards the company. I'm not biased towards anyone at the table. I will speak up and say no. There's tons of people like me. It's not just me. There's a lot of people that do this. It's our job. Like we're beholden to a certain guidance and criteria. To talk about the anything nefarious, I'd say be careful signing away your rights. Ask questions. If you are a part of a trial, ask what will, if I help this trial move forward, what comes to me? What therapy, like if I help this trial and I'm a part of this specific cohort that received this milligrams per dose and it worked, can I get access to that therapy? Like ask those questions. So be an advocate to yourself so that there aren't any nefarious players. I wouldn't say that where I sit, I don't get to see nefarious players because of the tone of where I sit. Like, yeah. And being a cop, no, I'm not gonna, they don't come to me, right, with nefarious things, right? So, but how I would protect myself when if I'm a participant, is I ask for access to the information, the publication, the guidances, any standard operating procedures, the Code of Federal Regulations. Like I know what to ask for, but I also ask, how is this going to benefit me, right? If I help you move your science forward, where do my cells go? Let's say I give a blood sample, what happens? Does it get destroyed? If it doesn't get destroyed, what happens? Who do I call if I don't want it to be circulated and sent somewhere else, right? And how do I verify that? So you have to be you have to be an advocate, and you have to stand up, you have to ask questions, and if they don't know, because a lot of people don't know, because I know what the sites know, because the biotech company trained them. But what the sites don't know can certainly come back to the biotech and pharmaceutical companies, because they know, right? So if the sites, the hospitals, the nurses don't know, you better believe the biotech people know. And they should go back and ask on your behalf, especially if they want you to participate in their trials. And I don't think I answered another question you had. I think I missed
I was just going to say a sort of continuation to that. And thank you for that answer. I just recently, well, not very recently, but fairly recently, was going to participate in a trial because I was very much wanting to benefit from whatever, healing could be offered from this trial. But what made me not do it was that they were asking questions about if they could just keep my information, my whatever they collected from me to use in perpetuity for many other studies for years. And I was like, no, I'm fine with this trial, but not in perpetuity for whatever. And what else would you be doing with it? And, some people maybe just kind of like, I looked through the contract. I read every single page. It was like 8 pages, but some people may not do that.
Right.
So I found that really interesting that they will use, if your consent for this trial, they will still, if you don't read, they could use it for other things as well, even not related to the study.
And you can always, what we all should know is that you can always, even if you sign that as a yes, you can change your mind the next day. You can always call and say, destroy those samples. And I've actually offended physicians by saying, I want that destroyed. Or they say, oh, I want to, I say, no. And I've had physicians, and I'm a whole person in the space, but the physicians who are outside of the space, but I've seen them where they're just like, oh, here's that lady again, but it's my data, it's my right. And if I'm not being compensated for it, yeah. So I feel, because you don't know what they're going to do. And I don't necessarily think it's nefarious, because sometimes they do need to know. Like if I have something rare going on, they do need participants to help them understand, how that disease works, like what therapies could alleviate symptoms. They do need participants, but you certainly can say, I'm okay with trial 1, 2, 3, 4, 5, 6, but I don't want to participate in XYZ. And that's your right. You can call every day, you can call every month, and you're still within your right to do that. And I recommend
for that.
Yeah. And don't feel like I negotiate a lot in life. This is just because I'm a negotiator. I love negotiating, by the way. But when you are having a difficult conversation, separate yourselves from any difficulty and just speak to the issue. And that's okay. Like, it's not even personal, right? I always tell my kids, like, when you see that stop sign, do you get mad? You just do what it says. You take heed, right? Like, we are always put in situations where we need to do something else, right? And so it's okay when you need a change and you speak up and ask for it, and there's nothing wrong with that. We're giving flags our whole life, and it's our job to heed and move forward. So definitely be your own advocate.
Thank you for this. I know you have to wrap up, but I appreciate you, giving us such a concise and just really, personal kind of like viewpoint perspective on her life, coming at it as a scientist yourself, seeing it from the other side and telling her story in this way and sharing it with us here for Black Metafest 2022. I really appreciate you putting together this exhibit. And if you ever want to do a reprise of this, again, so we have a wider audience, I would love to do that.
Yeah.
And maybe with a younger group of people. This Black Metafest 2022 is also about connecting people. So if I can bring your presentation to any local museums, I would love to do that, because I think this is just so important. She has given so much to the world, and we can honor her in this way by making sure everybody knows her name. So thank you. Thank you so much. Thank you so much, Leona.
Thank you. Thank you,
guys. Thank you, everyone, for coming. It's been an honor. I created a fireside chat area out here because if anybody wants to join me in the future, this space will be available. I'm so excited for the opportunity that you presented. I'm willing to discuss it for sure. But I would love to teach the youth about her and the world. So I'm good with that. And I love this space because the metaverse allows you to do it, right? And meet new people like Mr. Pierpont and talk about it. So, and I'm glad to meet all of you. Inspire the Goddess. I'm excited to meet you. Thank you so much for what you do, Soul Lady. I appreciate you. And of course, French. Thank you for everything.
Well, and I just want, if people who are interested in kind of arranging a presentation with you, can they reach you maybe on Altspace? Is that okay for them to reach out on Altspace or Clubhouse?
Of course.
As well?
Altspace, Clubhouse, Lyona Carter, Altspace, MetaLy, so Meta LY, or Metaly they call me, which is okay. And you have my email, French. You can reach to me directly as anyone.
Thank you so much. I just want to make sure people know how to, to kind of connect with you about this and giving a presentation, and I will certainly directly connect you. And thank you again, and thank you everyone watching in the Discord. Thank you so much for people who have joined us for Black Metafest 2022. Leona Carter, thank you so much for sharing your wisdom and experience. I appreciate you, and I hope you have a great rest of your day, and I hope to see you at some of the other events here for Black Metafest 2022. Thank you.
Yes, we popping out. I'll be walking in Crypto Voxels on Friday night in the fashion show, so I will be here.
Oh, yes. I can't wait to see that.
Thank you.
All right. Bye.
Thank you. Bye.
Bye for now.
Bye for now.
Hey there. How are you doing? I saw this on the list. I thought I'd come drop by. Oh, brilliant. That's so cool. I love that.